So we used seizures originally as a way to understand what we are doing, because the protein we discovered, its name is C-FOS, as a regulator of other genes. So we kind of discovered a conductor in the orchestra.
Fellow Homo sapiens, my name is Torie Robinson and welcome back to Epilepsy Sparks Insights. If you're new or haven't done so already, please do like and comment on this episode and subscribe to our channel so as to support our mission to decrease the discrimination faced by and improve the quality of life of those affected by the epilepsies, and get everybody appreciating and encouraging funding for the amazing epilepsy researchers out there. Now, many of us know that there are close correlations / links between loads of diseases, including the epilepsies, mental health disorders, including psychosis and schizophrenia, which aren't spoken of enough, in my opinion, intellectual disability, cognitive dysfunction, and way more. So that is why it is incredibly cool that I can today introduce you to the fabulous neurobiologist and research lead, Leszek Kaczmarek.
I work at the Biological Institute in Warsaw, Poland. It's called NENSKI. I established there a group in 1986 working on molecular neurobiology, trying to address questions of relationship between brain and mind, starting from learning and memory in animal models.
Can you tell us how this relates to the epilepsies, please?
It is rooted in this learning and memory studies, because what we discovered late 86 was that when rats learn new things, they can be the induction of a gene expression in the brain. But the first experiment was actually not even learning that the rats learn something, what is good, what is bad, but we injected into the brain a substance, neurotransmitter, that is critical for establishing changes in neural circuitry that underlie learning, and this is called glutamate. And then we wanted again to follow this, and putting something into the brain is complicated. You drill a hole into the skull and so on. That's not easy and nice. So our colleagues from Krakow, they showed us there's another way that you can inject the animals with an analog of the glutamate. It is called kaimate, that you can inject intraperitoneally, it means peripherally, not to the brain. The drug penetrates to the brain, activates neurons. The glutamate has to act on so-called receptors, so it activates glutamate receptors. Neurons get activated, spiking activity, electrical activity, and they release their own glutamate to activate other neurons. So it kind of perpetuates and results in seizures episodes. So we used seizures originally as a way to understand what we are doing, because the protein we discovered, its name is C-FOS, turned out to be, other people show this more or less late 80s the same time, as a regulator of other genes. So we kind of discovered a conductor in the orchestra, in the neurons, in the genomic, we say, response. So we wanted to see what are the genes regulated by this transcriptional regulator. And then we found a gene and documented that is regulated by this C-FOS, and the gene encoded protein called TIMP1, tissue inhibitor of matrix metalloproteinases. Then we asked, okay, what is TIMP1 doing? We never heard about that before. Okay, and it turned out that it's a protein that inhibits activity of some other proteins outside the cells, extracellularly, especially MMPs, matrix metalloproteinases, and among them MMP9, because there's over 20 and each of them has a number. So we follow with this MMP9. And since we started with seizures, and then we followed that as MMP9 is also strongly activated by seizures, okay, we just ask, okay, seizures, especially status epilepticus, intense seizure episode, may lead to epilepsy through epileptogenesis, the latent process of developing epilepsy. So let's see whether our MMP9 contributes to the epilepsy. So this was a paper published a long time ago, 2008, in which the most important finding was that we used mice that were missing a gene encoding this MMP9. It's called knockout. There are over 20,000 genes in human and mice, and only one, through genetic manipulation, was missing. And we showed that those mice are somewhat protected from developing epilepsy. So then we produced transgenic rats that have additional copies of a gene encoding MMP9, but only in neurons, selectively in neurons. And turned out that those guys are more prone to develop epilepsy. So at that time, we provided evidence that MMP9 contributes to epilepsy. And on the side, we still continued learning and memory experiment, etc., and actually showed that this MMP9 contributes to learning and memory as well. So in that sense, the link is a protein that is important. And there's a very simple explanation, which we have also provided. Why is that? Because learning and memory is this producing adequate responses, our responses, animal responses, to incoming stimuli from the environment. If we see that if those stimuli we met before, we were exposed to before, or similar we were exposed to before. And this exposure to the new stimulus for the first time somewhat reorganizes neuronal network that forms the brain. And this reorganization is called plasticity. By the way, this is the term introduced. It's very widely used. No, very many people know. That actually was introduced by Jerzy Konopski from the Nensky Institute in Warsaw in 1948.
Oh, I didn't know that. Okay.
I have to follow the giant and stand on the shoulders of a giant. So anyway, so we implicated this MMP9 in the plasticity. And what means plasticity? Plasticity means that the neurons are more or less responsive to other neurons inside the brain. Okay? And this happens, neurons are connected through synapses. These connections are called synapses. And the synapses may be of different shape and size. And what we found that MMP9 contributes to the modification of this shape and size of synapses. The excitatory so-called synapses, those synapses that respond to glutamate, are located, vast majority of them, on a small protrusions on the membrane of neurons on the part called dendrite. So they are called dendritic spines, like spines of rose. And then we found that MMP9 is critical for reorganization of dendritic spines. And epilepsy is also a disease that may have roots in this abnormality of synaptic plasticity. So there's a physiological synaptic plasticity leading to learning and memory, but maybe pathological synaptic plasticity that may lead also to epilepsy.
It's fascinating. And so tell us a bit more about the links between the epilepsies and other, mental health disorders. I've read that you've written about like schizophrenia and autism and cognitive dysfunction. What are the links between those things in your work?
In our work, MMP9. Because
it is still.
Sometimes those diseases are called synaptopathies. It means pathology of synapses. And we show that MMP9 is critical for the proper functioning, or maybe, when abnormal, to dysfunctioning of those synapses. So in that sense, we have implicated MMP9 in alcohol addiction, in depression, in autism spectrum disorder, as well as schizophrenia. We study mostly animal models, or even in vitro cultures of neurons very distant from the brain, one may see. But we collaborate with clinicians. And the studies have been really validated on the cohorts of patients, because the nature made an experiment on us. And this experiment is that the gene for MMP9 comes in various so-called polymorphic variants. They are not mutations, but small changes in the structure of a gene. And somewhat these changes may result in different levels of MMP9 production, including at the synapse. So clinicians study patients, study the polymorphism, and then we link the polymorphism, this variant versus this variant, to this and this symptoms of disease. For example, we found that MMP9 with Hannelore Eschenreich from Göttingen, we found that MMP9 contributes to hallucinations in schizophrenics. or with Jerzy Samochowiec from Szczecin, to greater motivation or lower motivation to seek alcohol in alcoholics.
It is amazing, really. And this is just one sort of protein, right? That has this impact that we know of.
What I'm saying here is that, okay, we have over 20,000 genes. The genes may code even for more than single protein, so we have tens of thousands of proteins. But in a way, so they build the cells. They make cell to be reactive to something. There are structural, there are enzymes, there are functional in various ways. But I would say that there are some are permissive to whatever activity cell performs, and some are instructive. Those are the rulers of the game. And MMP9 is one of those instructive proteins. So I think it's more important than others. But obviously I'm biased because I study MMP9.
Okay. Have you discovered anything that has surprised you or contradicted your presumptions about this gene or about the effects of this gene or what the causes of these diseases?
I'm not sure, really. It means that we are... Okay, my research, I'm saying neuroscience research started in 1986, and it sort of follows kind of results. We ask a question, and then we follow the result. So we are open to whatever comes. We don't make too many predictions like this. We ask specific questions. Does it contribute to epilepsy? Does not. So we may hypothesize that it does, but okay, it does. So it's not really very surprising. There are minor technical things that might be surprising, which is the time course of the activity and so on. This is a protease, so this is the enzyme, this is a protein that digests, cleaves other proteins. So understanding what are the targets, this was kind of a surprise, etc., etc. But fundamentally, I think I'm kind of happy that what I started in 86 is ongoing work.
That is science. I love it. I love it. What do you foresee the next 5 to 10 years holding for your research and the potential impact maybe on people affected by the epilepsies? I understand that, that you've already said that you are working with rodents and in vitro. What do you think the future may hold?
I am curiosity driven. When I was a young boy and was given a watch, my first thing was disentangle this watch. And I tried to put it together. Some parts remained. Sometimes it worked, sometimes it didn't. So I'm still like that. So I want to know how things are. So it's very fundamental in a way. But when I see potential for translation, application, I do not escape because it is also interesting trying to do this. So with this MMP9, the MM is an enzyme. Enzyme can be blocked by things called inhibitors. So enzymes can be inhibited. And there are many drugs that are inhibitors of enzymes. And actually, over 20, 30 years ago, MMPs, including MMP9, were targeted by potential therapies, by drugs that were inhibitors of these enzymes, because it's been proven that MMPs contribute to cancer metastasis. And this has been shown very well in animals, in this in vitro culture, and then they went to human clinical studies, even so-called phase 3 clinical studies, big studies. And everything failed. Billions and billions of whatever dollars, pounds, euro were lost because people suffered from the pain, musculoskeletal pain, because to prevent metastasis, they were given the drug chronically for a long time. So what we discovered, that this MMP9 is activated by those conditions provoking epilepsy, so-called acutely, within minutes to hours. Within a couple of days, MMP9 is entirely gone. The plasticity, this reorganization happened and may produce long-lasting effect. But MMP9 doesn't play a role. It's not involved in maintaining the effect. It just produces the effect.
Just like a trigger, right?
It is a trigger, but it's not involved in maintaining this to make it sustainable. So what happens is, I had this idea, I published this over 10 years ago. So maybe we should reconsider, it is called repurposing those drugs to some neuropsychiatric disorders in which MMP9 is triggered for a short time. And epilepsy is a prime example. And the conditions in adults to develop epilepsy are most often like traumatic brain injury, because car accident, exercise accident, or war thing, and so on, or stroke. 15 to 20 people after traumatic brain injury or stroke suffer from epilepsy that develops over like even a couple of years. And this process is called epileptogenesis, development of stroke. So we hypothesize maybe some of those drugs can be repurposed to prevent epileptogenesis, or at least slow down and so on. And what happened, so we had a project with colleagues with the company. Okay, let's take over 10 of those drugs and see whether they penetrate to the brain. Because brain is protected by so-called blood-brain barrier, it's not easy for the molecules to penetrate. And we discovered a couple that do penetrate. So then we tested whether they sort of slow down, prevent partially at least, a development of epilepsy in mice. And we found a drug that worked quite well. It's called Marimaster. Then I teamed up with a company, a small company established in Poland, the name is Picralida, that they decided to follow this. So we applied for patents worldwide. We sold them license to follow. Then we got a joint project with them, and we still continue to collaborate. And they had to make a formulation of a drug that would be suitable to humans. And they went all the way to so-called phase 1 clinical trials. It means whether the drug is safe in humans. And it proved to be safe in much higher doses that are needed to prevent development of epilepsy in animals. So what they are interested now, they have to find funds to go to clinical phase 2 trials to see the efficacy. So it is all the way still in progress. And hopefully, if you ask 10 years, why not? Could be a drug that would prevent development of epilepsy.
Thank you so much to Leszek for giving us what was really a rather scientifically, academically enticing insight into his brain-mind research, because I don't know about you, but I already want to know more. Check out more about Leszek and his research on the website torierobinson.com, where you can also access this podcast, the video, and the transcription of this episode all in one place. And if you haven't already, don't forget to like and comment and subscribe to the channel, and share this episode with your friends, your colleagues, your family members, university, schools, whoever, the whole shebang, because this supports our mission to decrease the discrimination faced by and improve the quality of life of those affected by the epilepsies, and get everybody appreciating and encouraging funding for the incredibly cool epilepsy research that is out there. See you next week.